Okay, thank you everybody. It's great to be here. We are Osteoleap Innovations and we make therapies for people with osteoarthritis. So before we get started, it's important to make sure everyone understands kind of the three major manifestations of OA. So OA is characterized by bony growths in and on the joint known as bone spurs, precipitous loss of cartilage, and subsequent pain. Now, this is a very common disease affecting over 500 million people globally and 33 million people here in the United States. It's also a very expensive disease, with the average patient incurring $14,000 in direct medical costs annually, and with a total joint replacement of the knee costing $30,000 at the hospital. Now, the issue that we see today is that current approaches really only treat the symptoms of OA. If you were to get a diagnosis today, you'd probably be given high-strength ibuprofen, maybe you'd be given a cortisone shot or other steroids, maybe you'd take glucosamine chondroitin and do some PT. Now, this provides short-term pain relief, but it doesn't get at the underlying structural issues of OA. Eventually, you'd probably deteriorate to the point where you need a joint replacement. These are major surgeries that require tens of thousands of dollars, months of physical therapy, and have a chance of infection or failure. What we're creating is something that goes against kind of these two approaches and creates a way that actually regenerates the joint itself. So, OA is a huge market. When we look at revenues of $5,000 per patient, we see that in the US alone, it'd be a $100 billion market. And obviously, that's way too big of a market for us to capture. But even as we think about going at kind of the younger group of people who are more likely to see the results of some of the regenerative properties of our drugs, we still see this market in tens of billions of dollars. So even if we capture even a subset of that amount, I think we could have a really good option here. Now, how are we doing this? So what we've identified is the bone morphogenic protein, or BMP, as a key player in osteoarthritis. And we've identified two different drug types, one small molecule and one larger biologic, both of which inhibit this pathway and essentially keep stem cells moving into the cartilage cell production mode rather than moving into the bone production mode, which kind of results in these bone spurs and a loss of cartilage. Again, the advantages of this is that we're actually able to repair cartilage in the joint itself and inhibit bone spurs and reduce pain, all without a need for surgery or anything like cortisone shots, something like that. Now, right now, what we're really focused on is further optimizing our lead. We really think we can create even better versions of the molecules we have today. We think we can extend the half-life so people need fewer shots, and we think we can also reduce side effects. We think this will take about a year and $2 million, and from there we'll be able to move on to studies in pigs and FDA-enabling studies, or IND-enabling studies, excuse me. Thank you very much. Appreciate your time, and I'd like to invite my co-founder, Dr. Weiguang Wang, to come up here while we answer questions.
Is it going? Ambitious plans. Do you start with a small molecule or biologic? Where's the focus?
So we've seen promising results in both. Given that, we think the small molecule is a better approach. It's going to be much cheaper, faster to market. It does have a slightly shorter half-life in the joint, so we'll need to develop a carrier or some kind of extended release mechanism to make sure that people don't need quite as many injections. And these would be injections into the joint itself, at least in the current formulation.
Any worries about the specificity of a kinase inhibitor?
I'm sorry.
Any worries about the specificity of a kinase inhibitor in that setting? I mean, those kinase domains are fairly similar.
Yeah, the 7 different BMP receptor. So based on the human global genomic study, using 800,000 human data, we found one specific BMP receptor associated with human OA. So we use the inhibitor specific inhibit this receptor. So this how we select the molecule.
But are the kinase domains of the different BMPs similar to each other and other kinases?
Yes, based on there's some similarity, but this specific BMP receptor, the function domain, the kinase domain, a little different from other member. So we can select it. Actually, people already selected before. We just picked the commercial available one, which had the highest selectivity compared to other kinase domain. So this is the best one when we're looking for it in the market.
Could you also just talk through how you propose to confine the therapeutic in the sort of knee joint? I think you said you
Yeah, it would be an
inject directly,
intra-articular injection into the knee joint itself.
And is there any worry of because you inhibiting bone growth, right? Which osteoporosis is an outcome of that. Is there any worry of sort of systemic effects, or do you feel that you can really localize it in the knee joint itself?
Yeah, we do expect it had side effect, inhibit bone, but that's why we inject specifically to the joint region. So the drug will stay in the joint because the capsule have very little vessel. So most of the drug, 90% of the drug will stay in the joint. And actually it turn over very fast because the small molecule had only 5 hours of the half-life. So it won't go to other tissue in the body.
Similar to the question that was brought up, one of your claims is that this is going to repair cartilage. So obviously, this is going to limit the efficacy of your product to patients whose cartilage needs repair. So if it's, for example, a bond, that this is not to rebuild the cartilage. Is that a correct assumption?
So I think we would need a little bit more longer term data to truly understand that, is the honest truth. But I do think that's part of the reason we're focusing on some of the younger age population, right? They're probably not so far gone that if we give them treatment now, we can actually prevent OA versus if you're 70 years old, 75 years old, it might not be worth it to do this treatment.
And the reason I'm asking that is that repair and rebuild is our two different, action. And if it's rebuilding, then to the earlier, then if it's rebuilding, are you worried that it might actually create bone spurs just as a side effect and kind of an off-target?
I'll quickly answer that. We certainly haven't seen that so far in our data. I don't know, is there anything else?
Actually, this inhibitor, it inhibits the BMP pathway. So it actually inhibits the bone formation. So we inhibit, because the stem cell can become bone or cartilage either way. So if we inhibit bone, then more cartilage is generated.
Yeah. So gentlemen, I saw you yesterday, which is why I'm not talking, but you guys did show a slide yesterday. It's not in this deck. You have it in your backup?
Yeah.
Because that's a lot of what Faranazo has.
Some of the results here. So this is some of the cartilage repair, right? So this is what you see after essentially 8 weeks of surgically induced osteoarthritis. And then on the right are these two different drug types. We allowed the OA to develop for 1 month, and then we gave treatment for 1 month. And there's an over, kind of 80% of the cartilage is still intact as compared to essentially close to zero. But of course, we need the longer term data to really understand how this is going to work over 6 months, 12 months, that type of thing.
I had a quick question regarding targeting using a kinase inhibitor versus just making antibody. Have you explored that? Seems like your option. You have an antibody, so, and I guess from a patent exclusive perspective, also you have an asset that possibly could longer. So,
yeah, actually, the biologic is FC protein, so it looked like working like antibody. It inhibits the ligand bind to the receptor. So the advantage of the biologic is they have long half-life, but the disadvantage is they're very expensive. When we talk to the CRO company to make it the GMP production is $5 million. So the small molecule only 50K or something. So we focus on the small molecule right now.
So, I mean, but at some level, you're saying you're making on a program based on your MNC cost drug product batch, right? I'm asking more mechanistically, like, does it make sense to develop the antibody? Because when you go at scale,
yeah, antibody, because the two different ligands bind to the same receptor, so you need two different antibody to block all the ligand. So FC protein may be the easy way to block both ligands.
Sure.
So,
Let's say FC protein,
Yeah.
but just using a biologic in general.
Yeah,
You're saying we made the decision based on your cost of drug manufacturing.
yeah, cost effective
Right.
right now.
But-
Yeah, yeah. So this is the only reason we focus on small molecule, but we didn't exclude the possibility to using the biologic, because the FC protein, the reason it's very expensive because it has a long peptide. The FC domain is big. So if we can cut out the FC domain, only use the actual cellular domain and link to some carrier, it may possible we can do, reduce the cost. We, for the optimization, actually, we work with both biologic and a small molecule. So we keep the opportunity open, actually.
Yeah, I would encourage you guys to look at the biologic route. There are several advantages to that approach, including specificity and likely also just life of patent and life of the drug.
Okay, thank you very much, everybody. Appreciate it.